Hey everyone, our next topic is regulation of the cell cycle and cancer. Well, we've already talked about the cell cycle and mitosis. Some things we need to remember. A multicellular organism needs to coordinate cell division across many different tissues and organs. And even with the problems with that, you have to think about timing and rates and the fact that not all cells are going to be on the same cell cycle. So there's going to be different rates, different frequencies of cell division depending on the type. An embryo may go through the whole cell cycle, including mitosis, in just 20 minutes. Skin cells, it may take 24 hours. Liver cells may not divide very often, maybe only every year or two or so, but at least they keep it in reserve that they can. Mature nerve cells and muscle cells, remember they permanently go into G0 and do not divide after maturity. Remember the different parts of the cell cycle. Most is interphase. You have the G1, the growth phase or gap 1 phase, synthesis of DNA, and then G2, then the M phase, which includes both mitosis and cytokinesis. All of that needs to be regulated. And we don't want any cells to go out of control. Any cell that divides out of control can become cancerous. So there's two irreversible points in the cell cycle. When genetic material is being replicated, once that starts, that does not get stopped. So that means we better make sure things are in order before that process actually begins. Another irreversible point is right at going into anaphase, from metaphase to anaphase. The separation of the sister chromatids, once that starts, you don't stop that. So again, you better make sure everything is fine within your cell before you start to divide those sister chromatids apart. So Mother Nature has given our cells checkpoints. This is where a process can be assessed to see if everything needed is there, or if we need to halt it because there is a problem. And then even after those checkpoints, can we fix the problem, or is there irreversible damage, and should we just give up and go into apoptosis? So those are some things to think about. On the bottom of this slide is a reminder of what chromosomes look like in their single strand fashion when they consist of just one chromatid, then in their double strand duplicated fashion, sister chromatids, and then when split apart again. Remember, homologous chromosomes come from both partners, like for instance chromosome pair number 1 or maybe the X's. Those are homologous chromosomes with each other, and they have the same genes on them. So the purple and the green chromosomes at the bottom of the slide are homologous. So let's talk a little bit more about these checkpoints. All right, there's three main ones. And so I've got our cell cycle here with the familiar labels on there, but then I've added the checkpoints. So these are some of the most important checkpoints. The G1 checkpoint, right during, more near the end of G1. Then we've got another one at the end of G2, and then at kind of the middle of the M, which is kind of interesting. It's kind of like a stop and go chemical signal, that critical points. These signals will indicate if the key processes have been completed correctly and whether we need to stop the cell cycle and fix things or go into apoptosis, or if we can just go ahead and all systems go. So at the G1 checkpoint, a cell might ask, well, can DNA synthesis begin? Because remember, that's one of those irreversible processes. If we begin DNA synthesis, we can't stop it. So we better make sure things are good to go before we get to synthesis. And that's what the G1 checkpoint is for. The next checkpoint at the end of the G2. Basically, synthesis has already occurred, so we can take a look at, did synthesis take place properly? Do we have too many mutations? Those are some questions a cell, if it had a brain, could ask itself. If things are good to go and we get the thumbs up, we're going to have a commitment to mitosis, and we're going to go ahead and go into that M stage. And then the third major checkpoint is the spindle checkpoint. This is right before anaphase, right before we're going to pull those sister chromatids apart, because once they start pulling apart, you don't stop that process. So again, we want to make sure everything is good to go before we begin that, such as are all chromosomes attached to the spindle. It is so important that when we pull the cell apart, that half of the chromosomes go one way and half go the other way, so that both cells will have the amount of DNA or chromosomes that they need, that they all have the recipe to make the correct proteins, and that no one cell has too many chromosomes, because it could be possible that chromosomes don't separate from each other and they end up going into the same daughter cell, and that's usually not good either. So again, we have our three major checkpoints at the G1, the end of G2, and the spindle checkpoint to help make sure that all systems are go. The G1 checkpoint is maybe our most critical checkpoint. It's like our primary decision point. In fact, it gets a special name called the restriction point. If a cell receives a go signal here, then we're going to commit to synthesis and usually division. So what are the signals all about? Well, these are internal signals like cell growth and size and nutrition, and external signals like growth factors. If a cell does not receive a signal, it exits the cell cycle and can go to G0, which is that harmless, non-dividing, non-working state. So that's right here at the G1. There's our restriction checkpoint. Remember the G0, these are for non-dividing cells like liver cells, nerve, and muscle cells. So let's look at activation of cell division. How do cells know when to divide? Well, now is when I want you to think back to chapter 11, to our cell communication chapter. Like an external signal, like a ligand, a hormone, docks in with a receptor on a cell, and think of the different changes that happen, the reception, transduction, and the response, all in our chapter 11. Well, that's what I want you to think about now. These are those chemical signals that give a cue that a cell should go through the cycle and divide. These signals usually mean that they're going to be proteins, and they're either going to activate certain things to happen in the cell, or it could also be an inhibition or an inhibitor. A neat experiment on the bottom of the slide. We have two cells sitting next to each other. One of them is in G1, just a growing interphase, and the other one is in M, and you can see that the chromosome there is a duplicated chromosome. It has gone through synthesis and is now in M. Now, what happens when we fuse the cell membranes of these two cells? Well, the G1 cell, which was right here, is suddenly acting like the M cell. And you can even see the chromosome in here, and it's ready to go through cell division, even though its chromosomes are not even duplicated. So what caused this? How can we explain this? Well, it's got to be those cell signal proteins that are in the cytoplasm, that once we fuse those two cells together, now they're sharing those signals, sharing those proteins, and those proteins must be activators that are now causing that G1 cell to do what the M cell is doing. It's all about proteins, all about those chemicals, these go-ahead signals. So there's protein signals that promote cell growth and division. and our internal signals. These are called promoting factors. External signals are more called growth factors, such as growth hormones. And the primary mechanism of control here is, ta-da, phosphorylation, which that's something we're all very familiar with, that we've been talking about for a long time. When you take a phosphate from a molecule, like ATP, a very common one, and you attach it to another molecule and it gets it all fired up and ready to do work. Remember, molecules, when they get phosphorylated, they get energized. So molecules that tend to phosphorylate others are called kinase enzymes. That word is not new either. We've been talking about that word for a while as well. So this phosphorylation can either activate or inactivate other molecules and enzymes within the cell. Let's see what other kinds of molecules that we've got to contend with here. So we've got the kinases that we just talked about. Then we've got these regulatory proteins called cyclins that we can see in the picture in the upper right-hand corner. And then these CDKs, that's short for cyclin-dependent kinases. All right, so we've got basically these kinases who are in the cytoplasm, but they're not doing anything until they get activated. You have to increase your cyclin levels, then the cyclin will dock with the kinases to activate them, and then you get this CDK cyclin complex, and together this is what's going to go phosphorylate other molecules to start triggering the passage of this cell through the different phases of the cell cycle. All right, so alone, these two molecules don't do much, but when you put them together, they get activated and then phosphorylate, these kinases phosphorylate things, and then you get the cell cycle moving ahead and moving along. Here's some scientists who were famous for figuring out the action of cyclins and CDKs and kinases by looking at the different levels of them in a cell and figuring out that when cyclin levels are high, then you get a high level of this MPF, which is called mitosis promoting factor, which kind of makes sense because cyclins bring about an activation of the cell cycle. You can take a look at in the notes. There's some videos you can watch to see these things happening. Here we can see where some of those molecules are active during the cell cycle, such as during G1, we have a CDK cyclin activated here. And at the end of G2, we've got another one activated, CDK cyclin, and that one's called the MPF, or mitosis promoting factor. And then during that spindle checkpoint, we've got some more active, although that one's called anaphase promoting complex. So it seems like these are molecules who are activated during those checkpoint places, such as at the end of the G1, the restriction checkpoint, the end of the G2, and then during right before anaphase. So CDKs and cyclin can drive the cell from one phase to the next in the cell cycle. So proper regulation of the cell cycle is so key to life that the genes for these regulatory proteins have been highly conserved through evolution. So this basically means that they really haven't changed much in no matter what organism you are. They're going to be very similar whether you're a yeast or an insect or an animal or a human or a plant. You all basically share these types of genes for these cyclins and these CDKs. Now there's some other things that can affect the cell cycle. Well, growth factors, that would be an example of an external factor, and there's some other external factors as well. So these are sometimes coordinated between cells who live next to one another, such as within tissues. So these are protein signals released by some cells nearby that will stimulate other cells to divide. So one of these examples is called density-dependent inhibition. So this is how crowded cells who live together within like a tissue stop dividing. It's kind of like they're talking to each other and they know that there's enough of them around and that there's no need to be dividing and making any more. So they release chemicals and such, or they can actually feel that there's somebody around them, so there's no need to divide. Then there's anchorage dependence, such as cells who actually need to be anchored to something or have their touch sensor receptors activated in order to go through mitosis or go through the cell cycle. So cells have internal regulators and they have external signals as well that regulate their cell cycle. So this picture should look pretty familiar. We looked at this in chapter 11 with how a growth factor signals a cell to start making proteins. So here we have reception happening with our growth factor hormone, also known as our ligand. When that docks here, then it sets up that different chain of events that happens. It might be like a G protein. Then we get our cascade or a transduction, which then basically activates a transcription factor to come on in here into the nucleus and then start transcribing this little gene right there. So it looks like we've got a CDK there who got activated. Back here we have some kinases who are doing some phosphorylation. Did you know that whenever you get a scab, that the platelets in your blood that helped cause that scab or that blood clot are actually releasing a chemical called platelet-derived growth factor that's stimulating the cells in that area for growth and healing? Basically mitosis? Well, now you do. Let's look at growth factors in cancer, because growth factors can actually create cancers when left unchecked or when left on all the time. So for our first one is called proto-oncogenes. These are genes that normally activate cell division. They become oncogenes or cancer causing when they get mutated. So we've got this gene who creates proteins to stimulate cell division, but it's under control and it's only activated when it needs to be. But if it gets mutated, it could get mutated in a way that it's on all the time. This can cause cancer. This can cause cells to want to keep dividing with reckless abandon to divide uncontrolled. One example is this gene called RAS. They tend to name genes. So this is called RAS. This one continually, when it's mutated, activates cyclins. And cyclins, as we know, make cells want to divide. All right, well, just because our proto-oncogenes are mutated there doesn't mean that the cell is going to definitely become cancerous right away, because we have some other backups as well. We have what are called tumor suppressor genes, and these are genes that inhibit cell division or shut it off. An example, in fact, a very important example, maybe one of the most important examples of tumor suppressor genes is one called P53. So if these genes are working, if a cell starts dividing uncontrolled, tumor suppressor genes can come in and shut it down. Stop them at their checkpoints, you know. But what if that gene also gets mutated in the cell, like the P53 gene? Well, now we've got proto-oncogenes making the cells turn on for mitosis continually. We've got the tumor suppressor gene not able to stop it and turn it off. So we can say this cell now has become cancerous or transformed when this occurs, when both of these mutations occur. So cancer is essentially a failure of cell division control. Unrestrained, uncontrolled, reckless abandon cell growth. And so generally, P53 is one of the most important genes in avoiding cancer, and so when we lose that gene P53 or it gets mutated, this is when we really get in trouble. We lose our checkpoint stops. Nothing to keep a cell in check anymore. If P53 is working, it can halt cell division if it detects something like damaged DNA. And then the cell could either stimulate repair enzymes to fix that DNA, to fix those mutations, or it could force the cell into G0, or halt it in G1 arrest, or if things are way bad, it could cause the cell to go into apoptosis, which is cell death or cell suicide. All cancers, in order to work, need to shut down the P53 tumor suppressor gene, meaning it needs to be mutated so that it no longer can turn cell division off. So P53 is kind of thought of as the cell cycle enforcer. So take a look at some diagrams here. Here we've got normal P53 gene, and we make those proteins normally then. Might include like a DNA repair enzyme. And when the cell divides, then we're fine. We can repair that. Or cell division could stop, and we could trigger enzymes to repair the damaged region and so on and so forth. Or trigger the destruction of the cell if it's damaged beyond repair. So P53 is enforcing up here, but here we've got a mutated or an abnormal P53 gene. And look what happens. If P53 gene is mutated, we don't make the normal proteins from that gene. So we don't have DNA repair enzymes, and we can't send our cell into apoptosis. So the cell can become transformed and then become cancerous. So basically what a cancerous cell is a cell that is dividing uncontrollably and making more of itself, and then it grows bigger and larger and starts growing into surrounding tissues and organs, could even move into the bloodstream and go to other areas as well. And when it does this, if it goes on too long, then it can stop your other organs and tissues from doing what they need to do if it grows too big. So literally cancer doesn't usually happen if it's just one mutation. Usually there needs to be about 6 key mutations for a cell to become completely transformed and growing into a cancerous tumor. So first you've got to turn your growth promoter genes on. So those are those proto-oncogenes that they're mutated, cancer causing, and they're on all the time with wanting unlimited mitosis. So you got to turn those on. Then for the tumor suppressor genes, those need to be mutated so that they're turned off all the time, and you don't make the proteins that fix bad cells. And then your cells can then ignore checkpoints and go right on through. Cells also have to turn off their suicide genes, meaning they need to escape apoptosis, so those genes must be mutated as well. They need to be able to divide with unlimited divisions, so they have to keep their chromosomes well maintained in order to do that. Normally, as a cell divides, every time it divides, the ends of the chromosomes get worn away a little bit. But in cancerous cells, they make repair enzymes to fix that. So it's like those cells are unable to age, and they will just live forever and continually divide. Also, all of the cell division takes nutrients and oxygen, ATP, and all that stuff. So they make enzymes to build blood vessels near themselves. They promote blood vessel growth. Tumors can get really disgustingly ugly with these massive blood vessels growing towards them. Then they need to overcome their touch or their anchor, that they could just be growing and they could break off into the bloodstream and continue to grow, that they don't mind that other cells are touching them and they're already in a crowd. They'll still grow and divide anyway. So they have to turn off those touch sensor genes. So what can cause all these mutations? Well, here's some things. UV radiation. How many people go tanning or spend too much time outside getting terrible burns? UV radiation can damage DNA, and all it takes is damaging the DNA enough in one cell. Chemical exposure and radiation exposure. Think of the things you come into contact with every day. Heat, cigarette smoke, pollutants, age, genetics, drugs, things like that. And then, of course, with the genetics, those are things you can't help. But some of these other things you can help. And of course, the older you get with age, the more you've come into contact with these things over your lifetime. And so the more likely that these different mutations can start adding up within your cells. So while you are young, maybe you only have in a certain cell, maybe you only have a couple of these mutations, but as you get older, you could get more and more of them. So tumors, like I said, cancer is like a bunch of abnormal cells, and if it's malignant or cancerous, it can actually leave the original site and grow into other tissues and organs. If it spreads to other areas, such as going through the lymph vessels or through the bloodstream, that's called metastasis. In a benign tumor, this one's interesting, the actual tumor or mass of abnormal cells remains as a lump at the original site. So this means the cells began to go out of control, but the tumor suppressor gene, p53, was still working. So it was able to halt those cell divisions and stop those cells from doing any more damage, and maybe make them go into G0 or suspend them in G1. Then a doctor needs to look to decide, can that tumor stay there or does it need to be removed? So this top one is a benign tumor, the bottom one a malignant tumor, and metastasis when it spreads. So how do you treat this? Well, one thing that all cancers have in common is that they are rapidly dividing cells. So the treatments generally attack rapidly dividing cells. So radiation is one thing, and chemotherapy drugs are another thing that attack rapidly dividing cells. Unfortunately, cancer cells are not the only rapidly dividing cells in the body. So cells like in the hair follicles and in your stomach are also rapidly dividing. So that's why someone might lose their hair, because those cells also get killed by the radiation and by the chemotherapy treatment. Also why someone becomes nauseous and vomits after these treatments, because the stomach cells are fast dividing cells as well. And then there are different drugs that pharmaceutical companies are working on to try to help find a cure or find treatments for cancer. This is one from Novartis called Gleevec to treat adult leukemia and stomach cancer. And this is a first successful drug targeting only cancer cells. We take a look at it here. Basically, remember when we had our enzyme chapter, remember inhibitors? That's kind of what it works like. Here's Gleevec getting into this enzyme and acting as an inhibitor, so ATP cannot get in there, and therefore it can't get phosphorylated, and then it stops cell division. Pretty ingenious, thinking about how molecules and body chemistry works and how that can be used to target cancer cells. The SciShow has a lot of awesome videos to talk about cancer. Why haven't we cured cancer yet? 6 common misconceptions about cancer. Those are all good ones to take a look at. I think most of us in our lives, no matter what age we are and how big our families are and friends, how many friends we have, I think most of us have been touched by cancer in some way, where we know someone who had it, or someone in our family, or a friend, or even I've had students in the past who have fought cancer. So let me know if you have any questions, and we'll be talking more about this in class.